Swayam Prabha. Digital India. Educated India.
In this lecture, we will discuss about protein tertiary structures, right. So, we have already discussed about the protein primary structure. What is the primary structure? Amino acid sequence. Then protein secondary structures, right. What are different secondary structures? Alpha helix and beta strands, right. So, then we derived several features or several properties from the protein sequences as well as for the protein secondary structures. In the previous class, we mainly focused on the different methods for predicting protein secondary structures. So, we discussed about statistical analysis, for example, Chao-Passman method. It is based on the propensity of amino acid residues at any conformation, either helix or strand, right, or coil. Then we discussed about information theory. It is called GAR method, right. They used the information theory and mapped the information at the central position as well as the window length of 17 residues, left side 8 residues and right side 8 residues, right, and use this information, right, to predict the secondary structures. Then we discussed about hydrophobicity profiles, the plot connecting the sequence and the values, right. So, either they get different patterns or you can take the average value and see the peaks to identify the secondary structures. Then we discussed about multiple sequence alignment, right. This is mainly based on the alignment of different sequences, right, in the form of either PSM matrices or any other different methods, right. So, they use this alignment for predicting secondary structures. Either they use the GAR method or they can use the ensemble based information from known structures. The later on, then we discussed about the machine learning techniques. Here they used all the information, right, and they train this information using machine learning techniques like neural networks or support vector machines or different methods, right, and to predict the secondary structures. We also discussed about the consensus method, right, the ensemble based methods or the meta servers, right. Either they take based on voting or train the output of different methods, right, to get the desired output. So, in this class we discussed on 3D structures, right. So, what do you think about the 3D structures? Which information we can obtain from protein 3D structures?
The
orientation as well as especially the you can say the coordinates. So, the tertiary structure or the 3D structures, so provide information about the 3-dimensional structure of a protein, right, atomic details. So, it can give you the exact location of each atom, right, in a specific residues on a protein, right, x and y z coordinates in the Cartesian coordinate system. Then coordinate structures, they provide the combination of the secondary tertiary structures having different subunits to give the tertiary structures. So, here if you see the protein, so you can see the structure of a particular protein here. So, there are so you can some of the secondary structures helix you can see in the form of spirals, and you can see some arrows here, they represent the strands, right, and then they interact with each other, the atoms and the residues, right. They interact with each other based on the various types of interactions depending upon the residue types. Either they make the electrostatic interactions or van der Waals interactions or hydrophobic interactions, and they form the unique 3D structures. When you talk about the 3-dimensional structure of a protein, it gives the atomic coordinates, say if this is an atom here denoted here. So, it gives the information regarding x, y and z coordinates. How to get the structures, right, how to obtain the information regarding the 3D structures, right. So, there is various experimental methods like X-ray crystallography, NMR spectroscopy, electron microscopy, right, the different methods which can help, right, to determine the 3D structures of proteins or other macromolecules, even for small molecules. So, if you look about the X-ray crystallography, it is one of the most prominent methods available in the literature. Currently, we have the very sophisticated instruments, right, crystallographic instruments, right. So, you can obtain the 3-dimensional structures, right, of even big big molecules if you are able to crystallize. In X-ray crystallography, the principle used in X-ray crystallography is what? X-ray diffraction, right. If you have the X-ray diffraction, right, this is the basic principle used in X-ray crystallography. So, there are various steps to use X-ray crystallography to get the structures using X-ray crystallography. First, we need to have a pure protein, right. So, in that case either you can synthesize the protein, right, or you can extract and then get the proteins, isolate the protein, right. So, and if you get the protein, then we have to ensure that this protein is pure. So, there are various steps which are involved in the protein crystallography. First you need to get the pure protein. So, protein purification. Once we get the pure protein, then we need to crystal, because the X-ray crystallography diffraction is important. In order to get a diffraction, we need a crystal. So, in this case we need to use different experimental conditions, right, so that you can make this protein in a crystallized form, right. What is a crystal?
Periodic arrangement.
The periodic arrangement of atoms, right. So, in the arrangement of atoms, there is the same unit, right, for all these units are the same, right. So, in this case you will get the crystallized of the protein, and then you need to next is crystal mounting is an also important step. Currently in several laboratories like the synchrotron addition and all, it is automated. So, in this case you can automatically mount at different directions and you can get the diffraction pattern. Otherwise, this is also an important step to mount the crystals in proper orientations and different proper directions. Once we do all these things, then you can pass the X-rays, right, and you can get the diffraction pattern. So, if you have the crystal, then you will get the diffraction pattern. This will give you what is the meaning of the diffraction pattern?
The position of the
This is the distribution of the electrons in the molecule, how they positions in the molecule. So, they are get diffracted, right, and then you can see the patterns. So, getting these patterns of the electrons, right, this we call as electron density map. So, you can have a get of maps, right, depending upon the population of the electrons in the crystal, right. So, you can get the electron density map. This is what we get using the diffraction of these crystals. Then after that we need to process. So, electron density map, right, this is location of these electrons, we need to process this data to get the structures. There are various methods to process this data, right. So, because we need to have the intensity or amplitude, right, as well as the phase. So, there are this is the problem with the X-ray crystallography. Now, there are different methods to solve the problem of this phase issue, right, and then you can obtain the structures. So, then once we get this process the data, we will get the structures. Then we need to refine the structures, because depending upon this quality of the structures, right, we need to match with this known data, right, known electron density map with known structures. Then they like to refine the structures, and finally you can get the refined structures. Once we get the refined structures, right, then this structure can be useful for doing an analysis to understand the behavior of these protein structures, what is the relationship between structure and function, where are the active site residues, which residues are involved in this type of stability, right, and folding and so on. This is possible. So, we see the X-ray crystallography, this is the major step. First we need the crystal, right, we have to get the crystal and get the diffraction, you can give a pattern like this, and we process this data, right, then finally you get the 3D structures, this is what we need. Then we check this quality of these 3D structures, right, and validating this using various methods, and one of the method is the Raman plot, and see whether these the residues are in the allowed regions, mainly in the secondary structures, alpha helices and the beta strands. So, we look into these known structures. X-ray crystallography is one of the most prominent methods, and you can see a quite large number of structures in the protein data bank that I will show very soon. And several researchers they got Nobel Prize, right, for using X-ray crystallography to solve the structures. Starting from the beginning, the discovery of these X-rays, and then using X-rays to diffract on crystals, right, and then for getting this for the small molecules to get the structures like the Bragg and Bragg. So, they showed that the path difference is the integral multiples of this wavelength. What is the famous equation for the Bragg's law?
N lambda equal to
2D sin theta equal to
n lambda.
N lambda, that is integral multiples of this wavelength. Okay, this is the path difference. Where theta is the angle between the incidence and the plane, right? And what is the D?
The interplanar.
D is interplanar distance, right? D is interplanar distance, right? Theta is the angle between the angle of incidence or angular refraction. So, lambda is the wavelength. So, they showed that. So, 2D sin theta equal to n lambda, and we get the highest diffractions only if these integral multiples of lambda, right. So, then they used this X-ray diffraction to solve the structures of proteins, mainly for the globular proteins, say myoglobin and the hemoglobin, this was solved structures, right. Kentu and Perutz, right, they got the Nobel Prize in 1962 for understanding the structures of globular proteins using X-ray crystallography. Then there are several other research like the vitamin B12 complex, and then they used for the DNA structures, right. Who got the Nobel Prize in DNA structures? Watson Crick got the Nobel Prize, and then they used protein DNA interactions, like got the Nobel Prize for the protein DNA interactions. Then for the case of membrane proteins, right, in 1988, Dyson-Hofer, right, Mitchell and Huber, they solved the structures of the first membrane protein, right, the photosynthetic reaction center, right, and they got the Nobel Prize in 1988. So currently in 2009, so we got the Venki Ramakrishnan, Tom States and Ada Yonath, right. They are 3 crystallographers, right. They enormously contributed to understand the protein production machinery, right, at the atomic level using the 3D structures of ribosomes, right. So they got the Nobel Prize in 2009. Also there are Nobel Prize winners for the GPCRs, right, and the other works on this protein crystallography. So X-ray crystallography is a major technique used to determine the 3D structures of protein molecules as well as other biological macromolecules. Then we have also NMR spectroscopy and microscopy are used to determine the structures. If you look at the NMR spectroscopy, right, it is based on the quantum mechanical properties of atoms, like the spin, that determines information about the atoms, right, and the local environment, right, with response to the magnetic field. So they have this spectrum and use the distance constraints in the NMR spectra and use these distance constraints to develop models. This is why if you look into the structures, for the crystallography they gave one structure, and the NMR they create several models, right. You can see the NMR structures, they have 10 models, and there is average structure from among all the different models, they get the average structures. So in 2002, Kurt Withrich, he got Nobel Prize for using NMR for 3D structure determination. So there are several techniques, and the major one is X-ray crystallography, NMR spectroscopy, and currently electron microscopy is also used for determining the 3D structures. So we solved all the structures, and all the structures are now deposited in protein data bank, right. PDB is called the protein data bank, right. All the structures are now deposited in protein data bank. In 1977, Burkina National Laboratory started to collect the structures of proteins. At that time there were only 8 to 15 structures, right. They gathered the structures and started to develop a data set from Burkina National Laboratories. Later on, when the structures are coming up, then they try to collect the more structure again and again, right, and they formed the consortium, right. They formed this research collaboratory in structural bioinformatics. They formed this protein data bank. So the protein data bank, so it is organized by the various institutions from different countries like Japan or the Europe or the US. So they have the PDB USA and PDB Europe as well as the PDB Japan. So they share the data from each other and they collect the information and they maintain the structures of these protein 3 structures. So now the growth of the PDB depends on the structures deposited in the protein data bank, right. Earlier days they accepted all the structures when they are published in the literature. So if you see the latest ones and the previous ones, latest one they have several options, right, and also they have to pass through several quality checks. And the older structures, they did not perform the quality check. There was several issues in the old entries. And secondly, earlier days the crystallographers or the using NMR spectroscopy, they solve the structures and they publish and then deposit in the data bank. The current scenario, first we have to get the code from the PDB before you publish your article. In this case, the PDB, right, who is maintaining the PDB, right, they curating the PDB, they check the quality of your structure, and if you pass the quality check, then only they assign the code. So the only after you get in the code, you can publish your research article. So in this way, they are able to maintain the quality of the structures which are available in the protein data bank. This is the reason why the PDB is the widely accepted the unique resource for getting the structures, 3D structures of proteins and as well as other macromolecules and complexes. So here I show the organization of the protein data bank and the data which are available in the protein data bank and how to extract the data and how to utilize the data from the protein data bank. So PDB is available in the RCSB website, right, Research Collaborator in Structural Bioinformatics. So they maintain the website, so which contains all the solved structures. So currently it is about, at the moment we have 1,33,589 structures, and these structures is increasing, right, periodically, right. So, every time you can see increase in the growth of the number of PDB structures available in the protein data bank. So, this is the front page of the protein data bank, right. You can see the statistics right here, and here you can see the search option. You can use any search option, right, to get the desired information from protein data bank. So, we see here there are several options available to deposit or to search. You can use in advanced search, right, and you can visualize the molecule, and you can analyze the molecule, or you can download, right, and there are several tutorials available in protein data bank which are related with the usage of protein 3D structures. Either there are several tools, or you can several analysis, or several aspects what we can do with these 3D structures of proteins. So, here you can use the search option, right. You can, if you want to have any specific protein, you, for example, this is the code for the lysozyme. So, 2LZM, this is a chain information. If you give this one, you will get this the data for this lysozyme. If you look into these statistics of protein data bank, okay. So, here you see, as I discussed earlier, X-ray crystallography is the most prominent technique to obtain the 3D structures of proteins. So, if you see among these 127,000 structures, so you can see about 114,000 structures are obtained from X-ray crystallography. Then if we see the NMR spectroscopy, there is about 11,000 structures, right, followed by the other techniques. And if you look into these structures of the different macromolecules, we see proteins, we can have the highest number of structures, it is 118,000, and about 3,000 nucleic acid, and the complexes about 6,000. This includes both protein RNA and protein DNA complexes. So, now you can use this PDB to get various information. You can get the collective information regarding the proteins from same families, or you can create non-redundant data sets, or you can get the data with any specific resolution, for example, high resolution structures or any specific organisms and so on. So, if you look into any specific code, for example, if you are interested in one protein, so I put 2LZM in the search option, right. You can see the search option here, right. We have put the 2LZM here, right, and then if you click on this go, right, then you will get the first page with the 2LZM. So, 2LZM tells this is bacteriophage T4 lysozyme. So, he give you the abstract of that particular protein. So, here left side you can see the structure, this is structure of the T4 lysozyme, and you can several options you can see here to display the file or to download the files. In the down you can see the method used, here you can see this X-ray crystallography and the subunits and different other details in the summary page. Then there are several other links, for example, you can see the 3D view or the annotations, sequence similarities or sequence information and the structure information, experiments and so on. So, if you enter the first page, you can have different options to get the sequences and structures. If you see here, this is the FASTA sequence, and I can also use the data for the full proteins. Either you get the header information without coordinates, or you can get the full coordinates. If you click on the FASTA sequence, then you will get the FASTA sequence. Okay, here this is the 2LZM, right, this is the FASTA format, right, and here you start the amino acid sequence from here to here, 164 amino acid residues, right, you can get the sequence in FASTA format. If you want to have the all the details of that particular protein, just you click on the PDB format, right. So, when you click on the PDB format, right, you can get the information. You start with the title and header, this is structure of this bacteriophage T4 lysozyme, and you get the more information regarding the source, and you can see the authors, right. So, Brian Matthews, okay, he is the one who solved the structure, and you give the reference journal articles, okay, they published in this article, right. Earlier we discussed about the PubMed, right. So, here you can see the full reference for this structure, right. Then you can see the remarks, okay, you can see the several other references and there are any specific remarks about the particular protein. So, if you go down to this page, right, so you can see the resolution, how far you can get the information about the 1.7 angstrom resolution here. So, what is the meaning of resolution?
The minimum resolution accuracy.
Yeah, accuracy you can get up to this 1.7 angstrom level, you can get the accurate position of these each atoms, right. So, earlier days we get with the high with low resolutions, say 3 angstrom, 5 angstrom. So, in this case the rectangular maps are not so clear. So, the position of the coordinates are also not sure, right. When we get more and more sophisticated instruments, Now you can get very clear picture. So even up to 1 angstrom resolution you can get the quality and the position of all the XYZ coordinates, right, with very reliably. So after that we go down, we scroll down the PDV structure, right. So you can see the residue, the sequence information. So these are the sequence information. Here the sequence are given a 3 letter code, right. This is M, this is N, this is I, this is F and so on. And then here then we give the data about the secondary structures, and if you look into the different PDV structures, some of them contain the secondary structures, some of them they do not have this information. And second aspect is the end residues are not very clearly given in most of these PDV structures. So it contains several helices, for example isolation at position number 3 to glutamic acid position number 11. So 3 to 11, so this can form a helix, right, from here to here, right, this helical segment. Right, then you can see the strand. So there are several helices here, this is about the helix, and you can see the strand here, right. This is the segments, right, for example is 56 to 58. So this is the isolation glycine 56 to isolation 58, right, this can form a beta shear. Here there are 13 residues in one sequence they gave, so accordingly you can see the location of the secondary structures. So now we go down, we scroll down the PDV coordinates. Now the next important thing, this is the important aspect of the 3D structures. What is important information you get from the 3D structures? Coordinates, right, you need the XYZ coordinates. Okay, this is the X, right, Y and Z coordinates. Again this is atom, right, for each atoms in the proteins or the nucleic acids. So what are normal atoms in the protein? Nitrogen, hydrogen, oxygen, sulfur, right. So you can see the NCO, right, and then you can see these atoms, right, N, C alpha, C O, these are the main chain, and C beta, C gamma, C gamma 1, C gamma 2, this side chain for this residue valine. For each atoms they give the coordinates. Here this is the atom number. So this is atom name, this is atom name, this is the atom number. So here you can see the residue name. Now this is the chain information, whether it is A chain or B chain. This is the residue number, and then we have the coordinates, the XYZ coordinates, and they give you the occupancy and the temperature factor, right, that I will explain very soon. So if you have these PDV coordinates, right, they give all the information regarding each atom, the atom name, atom number, residue name, residue number, and the chain information as well as the coordinates, X, Y and Z coordinates. So they also have some water molecules or some ions or some other ligands. If they are present, they give as heteroatoms. So you can distinguish the atoms and the heteroatoms, right, whether this belongs to protein coordinates or it is water molecules or any other ions or the ligands, right, they use to crystallize, that co-crystallize this particular protein. So now I discussed earlier, so we have the XYZ coordinates, right, and then you can see this is the occupancy, and I will explain what is the occupancy which are given in the protein structures. If you look into the here this term occupancy, most of the cases it is 1, and some cases you see there is a different numbers. What is the meaning of the different numbers and why this is 1, right, I will now I will explain. So what is occupancy? If you look into these macromolecular structures, macromolecular crystals, they are composed of many individual molecules to pack together in the symmetrical arrangement. So if you look into these structures, the side chain of the surface may just work back and forth, right, between several conformations. In this case, a substrate can bind at different orientations, either orientation X or orientation Y, right, in the active site, or the metal ion may be bound only few of the molecules. If in this case, when build the atomic model, they give the term occupancy to see how much each conformation is observed in the crystal, whether only one conformation is observed in the crystal for each atom or this atom can have different conformations. If the occupancy is 1, what is the meaning of that? So that atom is found in all the molecules the same place in the crystal. So there is no deviation. So almost all the molecules, right, this atom is present in the same position if the value is 1. In some cases, okay, this is the example for 1. So for all these, in these cases you can see the occupancy is 1. So all these atoms, right, these atoms in this residue, right, occupy the same position in all the molecules. So, this is why the occupancy is 1. In some cases it is different, say this one, this is the tyrosine 146 C beta, the same beta tyrosine 146. Here you can see the value, this is 0.49 for one case and another case it is 0.51. What is the difference? How it is 0.49 and 0.51? So, for example, if a metal ion binds only one half of the molecules, you can see the electron density map, right, and based on that they assign the occupancy. If it is same orientation, right, then they put the 0.5, right, in the both cases. What is the meaning of this occupancy? This will tell you the fraction of the molecules in different conformation. For example, two different conformation, right, I showed it here. So, two different values 0.4 and 0.451. In this case, this atom, right, the C beta of tyrosine 146, they have two different orientations. For example, this is 0.49, 0.51 and the total should be 1. So, there are different cases, for example 0.5 and 0.5 or 0.4 and 0.6 and any fractional occupancies, but total should be equal to 1. So, here I show an example. So, this molecule, right. So, they have the different conformation, this is glutamin 8, right, this is glutamin 8, one is occupancy 0.57 and the another one has occupancy of 0.43. This is another example, here this is a tyrosine 151, here both of them they have similar occupancy. So, in this case this is 0.5 and 0.5. If the same position is occupied by all the molecules, then the occupancy is 1, depending upon the binding. So, it may have different orientations. If they have different conformation, then the occupancy is split, so that the total will be equal to 1.